What? അറിഞ്ഞില്ല ജോമർ എത്ര Right. Good evening, people. Can you hear me? Do you hear me? Hello. Hello. Why you students? Can you hear me? Please send to the main chat. Yes. Yes. Okay. So today, people, we are going to discuss the plant form and function. Small revision we're going to do. Plus, we have to do photosynthesis and fungi. It's a bulk meal. It is a bulk, right? So we'll. Where do you want me to start first? Where should we start first? Should we start from photosynthesis, or you think photomorphogenesis? Which one is better, right? We'll start with photomorphogenesis, ne? We'll start with photomorphogenesis, right? Photomorphogenesis. Okay. So in grade 12, your resource book, grade 12 resource book, the plant form and function, we recently discussed this part. We discussed it very recently, plant spacing. So plant responses, right? Plant responses. We'll go quickly through that. Plant response is slow, I don't know why. Plant responses to internal and external signals. You can't read the letters, ne? You can't read the letters, ne? It's two, right? Plant responses to the internal and external signals. Plant responses. Right, people? Plant responses. So we will talk about plant responses we classified. Plant responses we classified. Plant responses to light. Plant responses to gravity. Plant responses to touch. Plant responses to not clear. This is not clear, that I know, right? Wait, just 1 minute. Light kadukarano me pen brighter ni one of pen it was right. People, we had to discuss a plant response to light, plant response to gravity. Background plant response to gravity, plant response to touch, plant response to hormones, and then we had to discuss plant response to stress, right? Plant responses to light is our first part. How plants respond to light. So how to change Zoom video quality? Not clear, sir, Mohammed. What's that? Zoom video quality you can't change. Ne? Right. That is what we stream, you can see. Ne? And I got in thought at the end. It been been. Right. So people plant responses to light. Plant responses to light. We can discuss different plant responses to light. What are those plant responses to light? Yes, the plant's responses to light. Photomorphogenesis, right? Photomorphogenesis. How plant form appearance is going to change people with the light condition. Photomorphogenesis we have discussed, right? And then the plant phototropism, bending of the plant paths towards light. Then plant spacing, and then flowering, seed germination. What is that list, people? So we have a lot of details here. Ne? The plant responses to light. We discuss, people, number one, photomorphogenesis. 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 And then we discuss, people, photomorphogenesis, phototropism, phototropism. Third one, plant spacing. And then people flowering, ne? Flowering, and then seed germination process, and then stomatal opening. Then there's another one we discussed, ne? What is that? Inhibition of the hypocotyle elongation, right? Inhibition of hypocotyle elongation. You send your chats to the main chat line, right? We have certain difficulties at the moment, right? The settings are not perfect. We had to buy a camera, right? So those are expensive, right? And until that, people will somehow go with the available facilities, right? What is the last one, people? Hypocotyle inhibition of hypocotyle elongation. Ned? Inhibition of hypocotyle elongation. Inhibition of hypocotyle elongation. Inhibition of hypocotyle elongation. Inhibition of hypocotyle elongation. Right. We discuss, people. We discuss these plant responses. We discuss the plant response to light. We discuss plant response to light. Right. We discuss plant response to light. Check this out, people. Right. We learn plant response to light. Plant respond to light, and then photosynthesis occur in plants. The photosystems are present, PS1, PS2, right? And then chlorophyll pigments are there, absorption spectrum, action spectrum, and all we discuss. So this is not regarding that, right? This lesson is not about plant photosynthesis. We are talking about how plant responds to light. To respond to light, people, there are two key photoreceptors available in plants. Ne? Plants are having photoreceptors. These are not pigments because they can't give any color. right? But they have photoreceptors, right? What is the difference? Photoreceptors are different to photosynthetic pigments. Why? Photosynthetic pigments provide color. So chlorophyll A, chlorophyll B, carotene, xanthophyll, what they do, people? They provide color to plant parts, right? Now these photoreceptors, which are helping the plant to respond to stimuli, they are available in low, concentration. So they are not concentrated to give some color to the plant parts. So they fail to give color, but they have the ability to respond to light. So we can talk about a particular action spectrum. Like photosynthetic pigments are showing some response. The efficiency of photosynthesis, change, people, depending on the wavelength of the light. So we talked about a particular kind of action spectrum there for photosynthesis. For plant responses, we can discuss a particular action spectrum, right? So you can see that these two plant photoreceptors, people, is this now, is the picture clear for you? Is the picture clear for you now when I screen share? Yes, neither. Okay, so don't worry about, yeah, we'll improve them slowly. Harida, until that people will go with the difficulty, right? These are like some old diagrams I drew, so I can use them still. So photomorphogenesis. We talk about a particular action spectrum in photomorphogenesis, right? So we can talk about two types of photoreceptors. What are those two types of photoreceptors, people? You can call blue light photoreceptors, which capture blue light. And then you can talk about, people, phytochromes, which are capturing red and far red light. So there are two types of photoreceptors present. Now, blue light photoreceptors are further classified as phototrophin. But in our syllabus, people, we don't talk about phototrophin. This is actually helping in opening of stomata, right? But we don't have to discuss about this. And then people, the second type we call as the cryptochrome, right? Cryptochrome. Crypto means, cryptocurrency, hidden. You don't know where they are. Same like that, we don't know where these cryptochromes present. Blue light photoreceptors are present on the cell membrane. And then phytochrome receptors are present in the cytoplasm. But we don't know where actually cryptochromes present. That is why we say crypto. So, cryptochromes are two types. Cryptochromes are doing two functions. One is phototropism. Second one is inhibition of the hypocortical elongation. It inhibits the hypocortical elongation. So we will discuss what they are, and then we talk about phytochromes. Now, phytochromes are photoreceptors. Phytochromes are photoreceptors. Phytochromes are photoreceptors. Now, these phytochromes are special because you have to understand, people, phytochromes are present in two stages, PR and PFR. Right? Phytochromes are present in two stages, PR and PFR. Right? So, phytochrome PR, and then it can alter between, people, PFR. So, PR, PFR, both are two status of phytochromes. It's like you are now at this stage. After some time, people, after having your dinner, then you're going to be like PFR, right? So you are ready to sleep. Sleeping mode one, PFR. PR to PFR, shifting can be done. Why you shift, people, from your present state? You're now active, conscious, awake. You can hear me. Then after the dinner, you are sleepy. So you transform here. With the dinner, you transform. The same you transform into a different state. Same thing can happen for PR when it receives red light. Red light, it goes into PFR. Ne? Then same like that, people. If PFR receives far red light, PFR can transform back into PR. So PR and PFR transformation is happening under people red light. What we said about the wavelength of red light, my dear? What we said about the wavelength of red light? Far red light wavelength 730. Neither. Far red light is 730. Red light is 660 nanometers. This is 730 nanometers. You have to understand that red light is useful for the plant because plant use red light for photosynthesis. So plant need red light. Why plant need red light? They do photosynthesis with that. Right? 660 is used for photosynthesis. So when red light pass through a leaf, 660 is absorbed by chlorophyll pigments for photosynthesis. So what remain when red light and far red light? Look at this, people. If this is a plant leaf, this is a plant leaf. Let's assume that this one as a plant leaf. This one is a plant leaf. This is a plant leaf. So when red and far red light both fall, right? I have to use a different color for far. Okay, I'll use the same color. Neither. Right? This is 660, 730. Neither. Both fall on the leaf. What comes down from the leaf, people? Only 730 comes down. Why only 730 comes down? Because the chlorophyll pigments available here. Chlorophyll pigments, right? Chlorophyll pigments are going to absorb. Chlorophylls. They absorb. Chlorophylls absorb light. Which light? Chlorophylls absorb, people, red light. So what remain is the far red. So far red comes down. So depending on this reception of the red and the far red, plant can show responses. So these phytochrome, they absorb these light and they can show responses. They show responses. So we can show when red light present. When red light present, what is available? PFR. It's like when dinner ready, you are available in PFR form. You transform into PFR form when your dinner is ready. Right? You transform into PFR form when dinner is ready. All right? So when you are in PFR form, then you are like sleeping. Right? When you have your dinner, when dinner is available, you are going to go into PFR form. So what happened with that? You're going to snore. Snoring. Goro goro budine. Right? Same like that. When plants come to this stage, people, plants show some responses related with that. What are these plant responses? Seed germination. Seed germination can occur. When the plant receives direct red light, seed germination can occur. Then plant spacing, that means, people, plants can branch, right? We say branching, right? In plant spacing, what happened? Plants are going to branch, right? And then, people, some plants, they do flowering. They do flowering, people. Flowering can take place. So therefore, when PFR present. So if you take far red light present, when far red present, PFR become PR, that means those three events are going to stop. So no seed germination when far red present. No branching, instead plants grow taller when far red present. And then, people, some plants, their flowering is going to stop. Now flowering is a special case, right? Flowering is a special case, right? Sometimes we can see that some plants, they flower in presence of red light. Some plants, they flower in presence of far red light. So now this is important. What is important? This transformation, PFR in dark, in night, right? Under darkness, what happens? Slowly, PFR transforms into PR. So with the dusk, with falling of night, plant available, plant has over the daytime, plant accumulate PFR. Then during the night, slowly what happen, people, they transforms back into PR. So in the early morning, with the dawn, plants are having more PR. So the moment they receive light, again they transform back into PFR. So throughout the day, plants are having mainly people PFR. Very short period time, their PFR level decreases and the PR proportion increases. So therefore, this ratio between the PR and the PFR is important for plants to respond. Is very important for plants to respond. Hope you understand this, right? We'll quickly go through your resource book, right? We'll go through quickly with your resource book. Right, so this is responses, plant responses to light, photomorphogenesis, key event. It will just give me 1 minute. I have to open this using a different app. Open with Microsoft Edge just once. So you can see my messages. Okay. That's the reason. I'll close that. This is the app. So I have opened with the wrong app. Oops. Right. So plant responses. So we'll quickly read your note for plant responses. Oh, right. Plant responses. Look at this. You read, huh? Photomorphogenesis, key events in the growth and the development of plants that are triggered by light are collectively known as photomorphogenesis. Key events in the growth and the development of plants. Action spectrum reveals that the red and the blue lights are the most important colors. Photomorphogenesis occurs with the involvement of two major photoreceptors, cryptochrome, phytochrome. Now, in our syllabus, they talk about cryptochrome and phytochrome. Okay, cryptochrome and phytochrome. And cryptochromes are blue light photoreceptors and phytochromes are red light photoreceptors. Okay, fine, neither. It was effects of light on seed germination. As the nutrient reserves are limited, many types of seeds, especially small ones. So what you have to remember? Many seeds, not the large ones neither. Small ones with very limited food reserve, people, they wait for the suitable light condition to appear for the germination. Otherwise, they don't germinate, right? So some plants are there, they wait until the light conditions become optimum and germinate only when the light environment and the other conditions are nearly optimal. Red light of wavelength 660 increase the percentage of seed germination. Far red light wavelength 730 decreases. Now, if you look at this, now people, seed germination. If you take seed germination, when the plant is growing under the shade of another tree, when the plant grows under the shade of another tree, right? Now look at this. This is the ground. Most of the seeds, people, you can bury them underground and they're going to germinate even without light. Most of the seeds can do that. Now, what we talk about small seeds with limited amount of storage materials, they are not going to grow, people, unless they receive the suitable light. Sometimes when there is a large tree and this tree canopy covers, and when the 660 and 730 light fall, 660 absorb for photosynthesis, so it does not come down. So what comes down is the 730. So the plant is now receiving 730. What is that, people? Farad. So when the seed is not buried, if the seed is present more superficially like that, the seed receives now 730. So the seed decide to germinate or not to germinate. Come on, answer my question. Do they decide to germinate or not to germinate? You have only one chance to answer, right? Do they germinate or do not germinate in process of 730? Come on, answer my question, Lamai. To the main chat. To the main chat. Come on. Yes or no? Yes, they do. That's 730. Come on. They grow or not? They don't grow. 730, no? Right? Do not grow. Suppose they receive, people, direct sunlight. Suppose they receive direct sunlight. 660. 660 is received. Then what happened? The seed germinate or not? Then they germinate. Then they germinate. So therefore, when talk about seeds, right? Seed germination is controlled by red light and far red light inhibits. All right. And then people, you have to also keep in mind that the seed, not only the seed germination, but also the hypocotyl elongation. Now, a seed has two parts. Seed has two parts, right? What are those parts of the seed? Parts of the seed. This is the seed. Seed embryo. This is the seed embryo. This is the seed embryo, right? So what I drew inside is a seed embryo. So it has two parts. Look at this green color patch, two bands, green color bands. What do you call these, people? You call this as the epicotyle. Epicotyle. The lower one, what do you call as the hypocotyle? Hypo means below, hypocotyle. Hypocotyle. Now this hypocotyle elongate during the seed germination. Hypocotyle elongate during the seed germination. So when the seed is buried underground, hypocotyle elongate like a loop. Hypocotyle elongate like a loop, right? And then pull the seed to the surface. Pull the seed to the surface. So this is the hypocotyle. Now they say that this hypocotyle elongation is going to be stopped. This hypocotyle elongation is going to be stopped as it receives what? Blue light. Blue light. As it receives exposed to blue light, it stops the hypocotyle elongation. So when talk about seed germination, both red light and blue light and far red light, they have effects on the seed germination, right? They have effects on seed germination. This is what it means, right? This is what it means. Look at these, people, right? Seed germination, red light wavelength 660, 730 during seed germination. When the seedling breaks the ground, light induced slowing of hypocotyle, elongation, right? Look at this. Light induced slowing of hypocotyle elongation is done due to blue light photoreceptors. All right, people, such seeds are often remain dormant for years until the light conditions become favorable. So sometimes some seeds are there, they don't appear from the ground until they get good light condition, right? Sometimes you do the same thing, until you get your requirements, you don't get up in the morning sometimes. Neither. Some of you, we know that. Until you get your requirements, you don't get up. So, same like that. Until the seeds receive their conditions, required conditions, they don't germinate. Right? So, they wait for some basic requirements. All right, people. So, then next one is people, plant spacing. Next is about plant spacing. Phytochrome provide the plant with information about the quality of the light, which enables the plant to adapt to changes in the outside environment. So this shade avoidance response of a forest tree below the canopy requires relatively high light intensity. Shade avoidance, right? Shade avoidance. Now look at this, right? Shade avoidance. When a seed is growing under the shade of another tree, it has to avoid and then grow. Avoid that shading and grow. Right? Am I too slow, people? Am I too slow? Just let me know. Right? You know that I'm teaching you the things that you have already learned, but it doesn't matter. You learn the same thing 2-3 times, people. It registers in your brain, mind, the easy way to learn. Right? So when a small plant is growing under the shade, what it receives now, we get 660 and 730. 730. So which one is going to receive down, people? 730 comes. So when 730 comes, plant decide to grow taller without branching. So the plant decide to grow taller to avoid the shade. So that is what you call as a shade avoidance response. It grow taller. But when the plant is growing, people, in open area, when the plant is growing in open area, the plant directly receives 660. Plant receives 660. So the plant decided to branch, right? So it decided to branch like this rather than growing taller. It's allocate more of its resources to branch. So you see this clearly in a forest with a canopy. Canopy is like tent-like upper, the crowns of the trees. The small plants try to grow taller to overcome the covering, shading. And then direct exposure result branching, shade avoidance response, right? So I think you understand that. Now there is a small error in the resource book in this part, but now people, it's not time to increase any errors in the resource book. We discuss these things in the class, right? What they say is the ratio between the far red to red light are directly thereby stimulating branching and inhibiting vertical growth. In contrast, exposure to the direct sunlight, increase of the proportion of the far red to red light stimulates branching and inhibit vertical growth. So just take it as a phrase and take it as correct, people, because if you try to discuss and then there is no point, because you know the reason. We correct it and then someone else correct it. It's given on that way in the resource book. So they're going to mark it as correct, right? So no point changing and arguing these things. We discuss them in the theory class. Harida. Ita passe lamai. Flowering. Right, let's talk about flowering. Photo. Ooh, what happened? Photo period is the oops. Right? Okay. Flowering, photo period. This is important. Good definition, Lamai. Can you write this as a definition somewhere? What is photo period? Right? What is photo period? Right? In 24 hours period in which the plant get exposed to light. Is photo period. Right? What is photo period? Right? In 24 hours time period, the plant exposed to light. How long that the plant is exposed to light is the photo period, right? Photo period controls flowering in many types of plants controlled by phytochromes. Okay. Then we talk about shoot elongation and phototropism. Shoot elongation and phototropism. Now, phototropism means bending of the shoot parts towards Bending of the shoot parts towards light. Phototropism. Now, when the plants are growing near a window, plant shoots are going to bend towards light. Plant shoots are going to bend towards light. That we call as phototropism. This is a window. You know this, right? When a potted plant is growing nearby, the shoot is going to bend toward the direction that the light receives. So we call this as the positive phototropism. Plant try to get more light. So the light in this case is now coming through the window. It comes through the window. So plants, according to the resource book says people, plants are responding. The plant responses are by phytochromes and blue light photoreceptors both. Maybe here phytochromes as well as blue light photoreceptors, both are participating here for the plant to respond and to bend towards the light. So that is important, people. So can you highlight that? Involvement, මොකක්ද involvement එක ළමයි? What is the involvement? We'll say that people, phytochromes and blue light photoreceptors both are going to involve in this case, right? Then people, response to gravity, right? Plant responses to gravity. Plants can respond to gravity. You know, people, some shoots of the plants grow upward and roots grow downwards. So gravitropism, right? Gravitropism. Now, plants' response to gravity, plant has some gravity sensing structures. You learn about them. What do you call these gravity sensing structures? And can someone tell me what do you call the gravity sensing parts? Which part of the plant actually perceives the gravity? Come on, somebody. Can someone help me here? Root caplu. That is Tahira. Paumi says root cap. That's very good, Tahira. Right, root cap. And yes, very good. And they are having statolith too. Statolith. And you know, statoliths are people, they are, what are they? How statoliths form? What are they actually, statoliths? What are they? They are form of starch stored plastids. They are something like chloroplast, but having too much of the starch stored in it, then you call them as a leucoplast. Neither. So they are kind of leucoplast. So what they do, they perceive gravity. Actually, the gravity reception is done by some different plant parts, especially at the seedling stage. Seedling stage. If you take this part as the shoot, and this part as the root. The roots are having special structures. The root cap captures the gravity. Root cap captures the gravity. Root cap here is having people those granules. What are these granules? We say people, the statolith, they are present. Though they can deposit from the direction that they receives gravity. And that is actually helping in distribution of auxin. Now, students, auxin is produced at the shoot tip by the shoot apical meristem. This auxin can transport all the way down, right, through the plant vascular system. They can reach here. And then auxin is going to accumulate. Auxin come and accumulate on the direction that the plant perceive gravity. Together with the action of auxin, and then plus people with the distribution of calcium ions also come and accumulate here. Auxin and calcium together suppresses the growth on this side. So what happened, people? This side growth reduced. Neither growth reduced. This side normal growth. The result is this is going to now bend downward direction. This is going to bend downward direction, right? Root is going to bend towards gravity. Now, people, something different is happening at the shoot. When the shoot receives gravity, they have not mentioned which part of the shoot is actually capturing gravity. No need. You can now there is a special part, but they have not mentioned that in the resource book. So you can just say that somebody is now telling it. Maybe the root tells the shoot, right? So the plant knows now from where the gravity comes. So what happened here? Auxin come and accumulate in the direction where the gravity perceives. What happened, people? Something amazing happens here. What is that? This side growth enhances. This side more growth, and then opposite side less growth. Opposite side has less growth. What is the result? What is the result? Result is this. It's now bending against the gravity. That's bending against the gravity. So there is a hypothesis, what you call statolith hypothesis. Statolith hypothesis. Let's look at that, people. Gravitropism occurs as soon as the seeds germinate. This ensures oh, board stucks. Board is stuck. Don't know why. Okay. All right. Statolith hypothesis. The aggregation of statolith at the lower points of the root capsules triggers redistribution of calcium, which causes lateral transport of auxin within the root. As a result, calcium and auxins get accumulated at lower side of the elongation zone of the root. At high concentration of auxin, cell elongation is inhibited. As a result, slow growth occur on the lower side and more rapid elongation of the upper side. Consequently, the root grows downwards. All right, people. Now we have to discuss plant responses to mechanical stimuli. I totally forgot about that part. Now we have to discuss about the plant responses to mechanical stimuli. Mechanical stimuli, touch specialist. Plants are touch. Some plants are touch specialists. They feel the touch more. Mechanical stimuli. So what we discuss, people, as the mechanical stimulations, we said thigmomorphogenesis. Some plants, if you go and touch them frequently, they don't grow tall, right? Some plants, when you touch them frequently, they don't grow tall. So you touch them daily, you go and touch the plant. Plant don't like that. They don't grow. So they stay shorter if you touch them daily, right? If you touch them daily, they can't grow. That is why, people, right? That is why in some areas with high wind condition, plants are like the fast blowing wind mechanically stimulate the plant to grow shorter. So when there is more mechanical stimulation present, plant fail to grow tall. So that is one of the plant response to the touch mechanical stimuli, right? Mechanical stimuli. And then, people, so what do you call this? Thigmomorphogenesis. Thigmo क्या है touch ने? Thigmomorphogenesis. Thigmomorphogenesis. Right? Thigmomorphogenesis. The second one we learn, people. Second one we learn, right? Plants can respond to touch, thigmonasty, and thigmotropism, right? So when a plant is growing around a support, think that there is a vine which is wrapping around the support and grow, right? When a plant grows around this sort of a support, when a plant grows around the support, you can see that there are two sides, neither. You can see two sides, this side and this side. One side is in touch. This side people touches. This side touches. That side grows slowly. Grow slow. Grow slow. The other side grow fast. The other side, the opposite side, grow faster. So that makes a difference. So this is going to move, wrap more and more around the support. When that happens, what do you call these people? What do you call this one? Come on, what do you call this? Thigmotropism. 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 Third type of speciality. What is that, people? Can someone tell me the third type now? This is thigmotrophism, thigmomorphogenesis. What is the third type? Come on, you tell me the third type. What is it? Yes, thigmonasty. Dinet says thigmonasty, all right. Now, thigmonasty means, thigmonasty is that mimosa plant response. They have pulvini. They have pulvini, neither. Pulvini, where the turgidity change at the pulvini when you touch, right? This is pulvinous. That's the pulvinous. And when you touch the plant, irrespective of the direction of that stimulus, the leaf is always going to show the same response. It's going to fall down. When you touch it, people, what happens then? The leaf is going to fall down, droop down like this. Right? Leaf is going to droop down like this. Leaf is going to droop down like this. Right? At the pulvinous, we can see that, people, the turgidity change, right? Turgidity change, right? Falling the leaf down when you touch, right? That happens. So this is what you call thigmo. What do you call that? Thigmonasty. Thigmonasty. That is thigmonasty, right? We'll quickly check your note for that part. Responses to mechanical stimuli, and the trees grows in windy environment normally have shorter, stockier trunks. The same species grow in normal environment conditions. Advantages of this could stand high winds. This exhibits the sensitivity of mechanical stress of plants. The changes in the plant form due to mechanical disturbance is called thigmomorphogenesis. Thigmomorphogenesis. During evolution, some plant species have become touch specialists. Climbing plants have tendrils that coil rapidly around support. Tendrils usually grow straight until it touches a support. The contact stimulates differential growth on opposite sides of the tendril. You know the tendril, right? Tendrils lamai. Tendrils kila kiyanne. You know, in passion leaves. This is the passion leaf. This is the passion leaf. This is that coiled stuff. That is what you call, people, the tendril. This is the tendril. Tendril. Sinhala, what do you call the tendril? Do you know the name of the tendril in Sinhala? Sinhala මොකද්ද කියන්නේ? Sinhala මොකද්ද කියන්නේ tendril එකට? Tendrils කියලා මොකද කියන්නේ? What do you call in Tamil? Huh? What do you call in Sinhala? Can somebody type it and tell me what do you call tendril in Sinhala? In Sinhala language, danne. Huh? Tamil learning hari kawanna kiyanna wal. What do you call in Tamil then? Oh, pahurak කියලා කියන නේද? පහුර සිංහල වීසේ පහුර. Right. Right. Pahurak. What do you call in Tamil? Someone, is there any Tamil? Pahuru. Kavisha said Pahuru. Tamil I'm asking. Kavisha. Tamil loli munadu kiyanne. You can say any line, no one knows, right? You can say anything. You can say anything, So you have to send your chats people to the main chat. I can't see the rest of the parts, right? You know, I have very Kasaru. Kasaru. And no name says Kasaru. Anonymous says Kasaru. Right. Anyway, good. The tendril. And then the directional growth of the tendril towards the support is called as thigmotropism. Other touch specialists respond to touch by rapid leaf movement. Example, Mimosa pudica collapse its leaflets when touched. Touching result in sudden loss of turgor. All right, you know the story now. You know the story. So we'll move to the next page. All right, plant growth substances. Plant growth substances. Right, now we have people, we discuss the terms here, neither plant growth substances, plant hormones, neither plant hormones. We learn people hormone word was the original term because the scientists were a bit curious to find hormones. The first type of hormone to discover was that insulin, right? That was insulin. And then they realized that somehow this pancreas is having some ability to control glucose. But there was no nerve connection, nothing. So they were like not neither knowing how it exactly happened. But anyway, they learn people some connection is maintained, secret connection. Neither. Sometimes secret connections, right? No one knows how you maintain the connection, right? But it's secret, right? The others can't understand. The same thing happened with the plant hormones. So the animal hormones, they realized and they thought that the plants are showing some similar behavior. So they thought, okay, maybe they are having some hormones too. But later on, people, they realize that even if they call as the animal plant hormones as hormones, the way that the animal hormones are working and way that the plant hormones are working completely different. So they wanted a new term, so they used the word plant growth substance, right, instead of plant hormones. And then later on, they found people not only the natural substances, but also synthetic ones are neither also responding, helping in controlling plant features. So therefore, they used plant growth regulators rather than plant hormones because the natural, synthetic, everything, right? So the plant hormones. So that is how the name came. So we drew a nice diagram, students. We drew a nice diagram for plant hormones. Let me show you people what I drew in Kurunagala class. Maybe some Kurunagala students are in the class too. Kohomada Kurunagala people? Kohomada, how is life? So this is your diagram. I'm showing them. Where is that? Maybe this one, no. No, not that one. I saved it. Okay, pictures, documents, pictures. Oh, yeah, it's here. All right, this is their diagram. We'll enlarge it, right? This cartoon guys, this cartoon people, So we discuss 30 functions based on these cartoons. 30 functions of plant hormones based on these cartoons. right? Very important to know them. Very important to know. So we'll take one by one, starting from one corner. Starting from one corner. Maybe this auxin guy, neither. Auxin guy. Can you see the diagram clearly? So the one at the corner, right? The auxin one. Auxin one, right? This one. I'm talking about this one, right? Auxins. So list of functions we discuss for auxins. Have a look once again, people. Have a look once again. Now pay attention for that. So stem elongation. Number one, first function, stem elongation. Number two, what's number two? Lateral and adventitious roots. Lateral and adventitious roots are going to form by auxin. Number three, fruit development. That's important. Fruit growth and fruit development, they are different, people. Fruit growth and fruit development are different. There was one question in today's 2025 paper, right? Fruit development and the fruit growth. What is development? Development is initial formation of the fruit, right? Then growth is, people, once the fruit, the basic structure form, it enlarges. That is a growth. So we can see a difference. So auxin involved in fruit development, not for fruit growth. Then people, number four. What's number four? Apical dominance. Apical dominance. Right? Apical dominance means what? Growing the apex. Apex grow faster. Number five, what is that, people? Phototropism. Phototropism means what, people? Bending towards light. Phototropism. Then number six, gravitropism. Number seven, vascular, xylem and the phloem, vascular differentiation. Vascular differentiation. And then number eight, this is people, stop abscission of leaves. Stop leaf abscission. Stopping of leaf abscission. Very important. Stopping leaf abscission. That is done by auxin, right? That is done by auxin. So we move to the second one. Second one is this gibberellin guy, right? We said that the gibberellin guy is like having all his thoughts for sexual reproduction. So you can see that, right? Stem elongation is an exception. Stem elongation, other than that, pollen development, pollen tube growth, sex determination or juvenile to adult transformation, and then fruit growth. All these things are like, people, events relate with the, those are events relate with the reproduction of the plant. So many events related to the reproduction happen in with gibberellin, right? With gibberellin. Then we discuss, people, cytokinin. Gibberellin, one more I forgot to mention that. What is this, people? He has a small child also, small child. So what is that, people? Seed germination, seed development and germination, both. Seed development and germination, both done by gibberellins. And then, people, next one is cytokinin. Next is cytokinin. Cytokinin. Cytokinin is a female, female cytokinin. Looks, so she, right? She, the cytokinin has functions, cell division of the root and the shoots, right? Cell division at the shoot tips and the root tips. Can you see this picture? Can you remember? Please find your notes. Hurry, right? Shoot, cell division at the shoot tips and the root tip. What is the second one, people? Second one is stop apical dominance and grow lateral buds. Stop apical bud. Now apical buds are now grow with the help of the auxin. Now cytokinin stops that and helps the lateral buds to grow. And then what is the third function, people? She works with the sink, female, so has some duty to do the cleaning and washing and all. So this is the sink. nutrients are transported to the sink. And then people, fourth one, seed development, right? Seed development is done by the cytokinins, and then stop senescence. So that is like somewhat antagonistic, neither. Stop senescence. Senescence is actually promoted by ethylene. Antagonistic function for that is done by cytokinin. Leaf abscission is done by, sorry, I made a mistake. Abscisic acid. Senescence promoted by abscisic acid, inhibition by cytokinin. Auxin inhibits abscission, then the ethylene promotes abscission, right, people? So then we move to the next person, right? This is the next phase of the life, ne? So we said that juvenile, young, it was a juvenile, and then people, this is coming to little middle age, the plant is now at the middle age. Now this is abscisic acid and its effects on the plant, right? Seed number one, what is that? Inhibit the growth. So this is inhibiting growth. Number two, what we said, people? Stomatal closure, closing stomata. Seed, number three, seed dormancy. Number four, leaf senescence, promoted. Then number five is something better, desiccation tolerance. By closing the stomata, this helps to tolerate the desiccation. Helps to tolerate desiccation. Desiccation tolerance is done. All right, that's abscisic acid, my dear. And then come to the last phase. Then come to the last phase. What is that, people? This old chap, ne? This is an old chap. Now he has coming to his elderly stage. So what do you see here now, people? Yeah, fruits are now ripen. Fruits fruits are ripen. Number two, leaf abscission take place. Number three, seedling triple response. Seedling triple response. Seedlings are going to leave now. Neither small seedlings, they show the triple response. What is actually triple response, people? What is meant by triple response? When the seed is stressed. When the seed is stressed. Why a seed is stressed? When the seed is growing underground, suppose it finds some obstacle. Seed is now developing, seedling is now growing. It finds some obstacle like this. So it can't grow taller because there is a rock. So what happened now, people? Plant increases the stress. So that means the plant produce more ethylene. When ethylene produce, this vertical growth is going to stop. And instead, people, horizontal growth is now promoted. And then people, the stem become more thicker, neither. More thicker. Stem become more thicker like that. So therefore, there are three responses. Triple response. Stop vertical growth. Promote horizontal growth. Increase, people, the lateral expansion, right? So it says the promote triple response, inhibition of the stem elongation, promote lateral expansion and horizontal growth. We call this as triple response of the seedling. So this cartoon is showing you, this cartoon shows you the triple response. Some of the kids are going to leave this guy. And then people, fourth one we said senescence, number four. Number five, people, root hairs are going to form. And then number six, people, flowering in pineapple family. Mala pine flowering in pineapple family, right? Pineapple flowers jump. Anna simal pine nenikammal. So those are the events, people. We discuss about the plant hormones, right? I want you to go through the notes. Put up balanda the podi post rakkem hadagan. Me balanda, right? So me sati, I'm going to give you people plant aramanada plant cell cycle poster kadena me juniors letter, right? You're going to get that poster too. Poster look right? So do something, because those are the learning tricks, people. I know that mitosis, meiosis are little different lessons for you. So get the poster if you want, right? On demand, we'll give you. If you don't have any space to paste them, no need to collect, right? We give it to the juniors. Right, people, we are moving now. We are now moving to the next theory part. So turn to next page, people. We have to discuss people plant stressors, plant stress, plant stress, plant stress response, plant stress. All right. Right. Plant stress. Right. People look. Plant stress. So we talk about people, several different types of plant stresses, right, people? What are those plant stresses? We talk about biotic stresses and we talk about abiotic stresses. What is, of course, a plant stress? Look at these people. A certain factor in the environment may have potentially adverse effects on plant survival, growth, and reproduction. Right? Certain factors in the environment may have potential adverse effects. It's like someone is threatening you. අවුෙන්ඩ එපා කකුල් කඩනවා හරිද අතපය කුඩු කරලා යවන්නේ හරිද අවුෙන්ඩ එපා පාරෙදි right if someone says like that neither then you get stressed same like that this is a stress for the plant හරිද certain factors are now putting plant life in danger we consider them as plant stressors we can classify stressors how do you classify stressors people So will you provide for online students as well? Oh, online එකේ ළමයින්ට දෙනවා කොහොමද? We'll post it. So abiotic stresses. Abiotic stresses ළමයි. Stresses we can classify into three ne. Abiotic stresses and biotic stresses. Abiotic stresses and biotic stresses. Abiotic stress and biotic stress. Right? Plant stress response. Stress response. Biotic stress. Biotic stress. Stress. Plant stress. Plant stress. Very stress. Oh, where is that? Okay, no need. It's not there. The picture is missing. Right. All right. People, plant stress. So we can classify the plant stress, abiotic stress into three. Ne? What do you say, people? Drought stress. You know that, ne? Drought stress, salt stress, cooled stress. Right? Drought stress. Drought stress. Now, some plants, when they are stressed, lack of water, the leaves are going to roll. Ne? Then we discuss another response shown by plants for drought. What is that, people? They close stomata. So, drought stress, if you take plants, plant stress, plant stresses, first one is drought stress. Ne? Drought stress. So, we said two responses for that. Close stomata. And then second one, people, we said roll leaf. Roll leaf. Leaf roll. Like in grass and all, they roll the leaf. Second stress response. Second stress response, people, is the cold stress. How plant responds to cold. Now, we learn, people, cold stress. When the cell membrane cools below the critical temperature, then plants develop the cold stress. So, what happened under cold stress? Cold stress. Now, in a cold stress, not freezing, cold stress, the lipids present in the cell membrane can solidify. When the lipids solidify, what will happen, people? Transport of things across the lipid bilayer is going to be slowed down. So, therefore, they increase, people, unsaturated unsaturated proportion of lipids, right? Unsaturated lipids proportion, unsaturated lipid proportion increase, ne? Unsaturated lipid proportion increase. That is, people, cold stress. Sometimes freezing is a cold stress, ne? They say freezing is another cold stress. Now, what happens in freezing, people? The cytoplasmic water tend to lose. So, to stop that, the some of the solute content present inside the cytoplasm is going to increase. Ne? Solute content in the cytoplasm is going to increase, right? So, that is another stress response. Read that part, people. Freezing is another cold stress response. Freezing is another cold stress response. And another cold stress, water in the cell wall and intercellular spaces freezes before freezing the solute rich water in the cytosol. The reduction of liquid water in the cell wall lowers the extracellular water content. So what happened here? The plant, as a response, they store move solutes in the cytoplasm. Okay, this is solute in the cytoplasm, which is harmful and may lead to cell death. Yeah. So what happened before onset of the winter? Frost tolerant plants increases cytoplasmic levels of specific solutes, sugars that helps to reduce the water loss. Right, that is important. Eh? All right, people. Then we come to the next plant stress. What is that? That is salt stress. Salt stress. So third one, people, salt stress. Right? Salt. I'll take plant stress abiotic. Salt stress. So what happens in salt stress? When the soil has too much of salt, plant fail to absorb water, right? This difficulty of absorption of water from the soil is now creating a stress in the plant. So we call this as the salt stress. So what the plant does, right? In mild salt stress, the cytoplasmic levels of the non-harmful solutes are going to increase. Mild stress, mild condition, non-harmful solute level in the cytoplasm increase, right? Non-harmful solutes increase. Non-harmful solutes increase. When very high salt stress condition, halophytic plants growing in people high salt, what is the response that they show? Right? What is the response that they show? They are showing people what? The salt glands. They develop salt glands, and then eliminates people this extra salt from there. And then finally, people becomes to the biotic stressors. Biotic stress, right? Biotic stress. Biotic stress are due to living agents. People can create stress on plants. Living agents, we call them as the biotic stressors. It can be sometimes bacteria, fungi, insects, whatever. They can cause people stressors on the plant. So we can understand people two types of biotic stressors. What you call the pre-existing, some sort of responses are there as a defense. Previously, now think that people, your home, you have some security, wall, the CCTV, dogs and all, they are pre-existing. You don't set them because you are under attack. You just set them up. That's for the defense. So same like that, plants are having pre-existing. And then some, we say people induced. When there is certain incident happen, then you quickly do this and that, and then you try to improve the security. That we call as the induce. So plants are showing both. Plants are showing both. Look at this, people. Pre-existing. Amount and quality of the wax and the cuticle cover the epidermis. Structure of the epidermis, cell walls and thickness. Size, location and shape of stomata. Thorns, picks and trichomes. Now this chemical thing is more important. I mentioned that long list of chemicals such as cyanogenic glucosides, alkaloids such as nicotine, phenolics such as flavonoids, lignin and tannins. So what is that? Nicotine is an alkaloid. Flavonoids, lignin, tannins are phenolic substances. Terpenoids such as azadirachtin and lectin, terpenoids. So these are different chemicals. Terpenoids. Now you have to pay attention here. You have to pay attention here. This is a part that you have to study. Very important part. Highlight that when you must remember this information. Right? What are phenolics? What are terpenoids? Alkaloids. They might ask questions regarding that. So you have a job to do. Memorize them. Okay? Then we will induce structural defenses. Induce ones means as the attack progress, some of the induced ones present, right? Morphological changes in the cell wall, formation of coke and abscission layers, production of phenolic compounds, toxic phenolic, only phenolic, ne? Look at this, people. Common one is phenol. Phenol, you can see people in the induced and as well as in the pre-existing defense. That's very important point. So you don't have the terpenoids, right? And then you don't have people alkaloids. What is common? Phenolic substances. They are common for both. Then production of toxic compounds. Some enzymes, they are also present. Some enzymes are present. So that's the plant part. That's all. That's all. That's all about the plant part. That's all about plants. Oh yeah, that's all. That's all, right? So plant part is done, right? Now people, we have very short time. I have taken almost 1.5 hours. We have to discuss quickly the fungi. Shall we have a quick look into fungal life cycles? Right? Oh, are you going to study it by your own? Photosynthesis I can't do today. Maybe I'll take it on another day. Photosynthesis I'll take on another day. Right? So go through fungi lesson also. Right? I'll take some other time and I'll discuss the fungi with you. Right? So, people, we discuss this part. We discussed this actually long time. We took nearly about people 2-3 classes to discuss this part. So we just did it in 1.5 hours. That's because people, you have already studied and you are quite familiar with the content, so we can speed it up. So I'll wind up for today. Right? Sorry about I failed to, because it's uh, my expectations are bigger, right? So then thank you very much for your participation. Hope to see you people in another class. Anyway, we'll be discussing the paper next Wednesday. I want you to answer it. Go through the fungi part, short part. Quickly balagandlamai. Harida. Lakoda amaru nani. Balanda. Thank you very much for joining tonight. I'm going to see you on Saturday, right? 1 o'clock. ආපු නැති උන්ට අද කට් කරපු අයට පේපර් එක ලියපු නැති අයට right මම තෑගි දෙනවා. I'm going to give them gifts on Saturday, right? So then see you all. Bye. Taggy.